Good morning, students. In the previous class, we were discussing about the complex permanent tissues. There we discussed why Xylem and Phloem are called as complex permanent tissues. And we also had the discussion that why they are also called as vascular tissues. After that, we took up the discussion on xylem. What are the cells there in the xylem? And how the xylem is different from other tissues. What are the main water conducting elements of the cells there in the xylem? We discussed about all those points. Even we discussed about the structure of tracheids, structure of trachea, even lignification patterns in trachea or vessels. All these points that we discussed. Today let me have the discussion over the third kind of cells there in the xylem. Xylem parenchyma. As far as xylem parenchyma cells are concerned, these are the only living cells of xylem. Okay, of the four types of cells present in the xylem, only xylem parenchyma are the living cells. All other kinds of cells which are there in the xylem, such as trachea, tracheids, even xylem fibers, they are all dead cells. And these xylem parenchyma cells, they don't have the definite shape. The shape is going to vary. Okay, the cells may be rectangular. Okay, the cells may be oval. The cells may be spherical. Okay, they vary in their shape. Suppose, say for example, if it is the xylem element, maybe the vessel, assume that this might be the vessel, just assume, okay? Associated with the vessel, we will find the thin walled elements, thin walled cells. Okay, these thin walled cells are supposed to be the xylem parenchyma cells. Not of that they are parenchyma cells only, but these parenchyma cells are associated with the xylem elements such as vessels or the tracheids. Suppose in the cross section, when you look into the xylem, the cells of the xylem more or less like this. Assume that these may be the cells of the xylem vessels. Associated with these xylem vessels, you will find the thin walled parenchyma cells. These thin walled parenchyma cells you can see. They may be either oval, sometimes they may be spherical. Okay, they differ in their shape. Various kinds of shapes can be observed there in the xylem parenchyma. Okay, these xylem parenchyma cells at the later stage, often they are going to develop lignin thickness. Okay, very slight lignin deposition may occur. Assume that this is the xylem vessel. Okay, after some times what happens is here lignin deposition may take place. Once the lignin deposition is going to occur, what happens is after the lignin deposition, the thin cell wall present there in the xylem, it becomes usually thick. Okay, these are converted into what we call as the thick walled cells, almost like this. And often these xylem parenchyma cells are going to develop what you call as the pit-like structures. There are some pit-like structures. Okay, these are supposed to be the pits, and these pits are formed because of low lignification. Suppose if it is the xylem parenchyma cell, just assume. Okay, here lignin deposition is going to occur, leaving some places, maybe like this. At some places there may be no lignification. Say for example, here lignification may not occur. Wherever lignification is not going to occur there, pit-like structures are going to be formed. We refer them as the simple pits. Usually simple pits are common there in xylem parenchyma cells. And these xylem parenchyma cells are mainly concerned with what you call as storage function. They are the storage cells. Storage of food present in the form of starch. Sometimes storage in the form of cannons. Sometimes storage of what you call as the gastric waste. They are all going to occur there in what you call as these parenchyma cells. This is with reference to that of xylem parenchyma. Then let me consider the last type of cells which are there in the xylem. We call them as xylem fibers. Okay, as far as these xylem fibers are concerned, they are usually the sclerenchyma fibers. Means they are the sclerenchyma cells present there in the xylem tissue. But these sclerenchyma fibers are present along with trachea or tracheids. That is why they are referred as xylem fibers. Suppose if you consider a xylem element, maybe the vessel, assume that this might be the vessel. This might be the vessel, okay. This might be the xylem vessel. Here you will find the cells which are very much elongated and having the tapered ends, just like this. These are the thick walled, elongated cells with the tapered ends or tapered points, just like this. This is the xylem fiber. Suppose if you observe a single xylem fiber, it more or less looks like this. Okay. This is one xylem fiber that you can see. Now, make out here. The xylem fibers are possessing all the properties of sclerenchyma fibers. They are dead cells, they don't have the living photoblast. Even they are thick walled cells. Walls are usually thick because of liquid deposition. Even they do exhibit what we call as the pits in their cell wall. And the cells are very much long, that is why we call them as elongated cells. And they are having the tapered ends means at the ends they are pointed. Suppose if you take the xylem tissue in the transverse plane, the cells that you can see like this, back out here. Okay, more or less, the vessels that you are observing are usually in spherical shape. Associated with the xylem vessel, sometimes you can see the polygonal shape, thick walled cells. These are the polygonal shape, thick walled cells. Okay. These polygonal shape, thick walled cells are usually referred as what? They are usually referred as sclerenchyma fibers. They are almost equal. be the vessel. These are supposed to be the fibers. Okay. These are the spherical kind of fibers. But here they are present in association with the xylem elements such as vessels. Okay, these are all vessels. Okay. In association with the vessels they are present. That is why we refer them as xylem fibers. But these xylem fibers, they are mainly meant for mechanical support. They give rigidity to the plant body. Whenever more number of these xylem fibers present, at that time the tissue becomes rigid, the tissue becomes strong. The stronger tissue usually offers mechanical support to the plant. That's why in xylem tissue, it is not only a water conducting tissue. In addition to the conduction of water, it also provides what you call as the mechanical support. This is with reference to that of what you call as the xylem. In your annual examination, theory examination, questions may be asked on this. Questions may be asked for 2 marks as well as for 5 marks. Okay, questions can be asked like this. Questions for 2 marks. Write a note on. Write a note on. Okay. Vessels, tracheids, xylem fibers, xylem parenchyma, okay, like this they may ask. At that time you have to explain each one of these in 2 to 3 sentences. What are vessels? How they looks? Okay, and what is the important function of the vessels? Like that, with each one of these you have to write 2 to 3 sentences. Okay. Even 5 mark question can also be asked on this particular topic. 5 mark question can be asked like this. Write brief note of xylem. Write a brief note on xylem. At that time you have to draw the diagrams related to the vessels, tracheids, fibers, parenchyma, and each one of the cells present in the xylem that you have to explain. Even instead of asking brief note, even they can also ask, describe the structure of xylem tissue. One and the other side. You have to describe all these elements of the xylem. Now I will move to the next tissue, that is phloem tissue. Write in the heading. Phloem. In other words, phloem is also called as bast. Bast is the other name of phloem. It is mainly a food conducting tissue. Conducting tissue means transporting tissue. It transports the food materials from leaves to all other parts of the plant. It is one of the complex tissues. When we are referring to the term complex tissue, it is clear that this tissue is made up of more than one type of cells. Then the phloem you can observe as many as 4 types of cells. The very first type of cells which are there in the phloem are supposed to be the sieve tubes or sieve elements, you can say. Sieve elements. The second type of cells present there in the phloem are companion cells. The third type of cells are phloem parenchyma cells. The last type of cells are phloem fibers. Of these four types of phloem cells, the first three types of phloem cells they are usually living in nature. These are the living cells. Whereas phloem fibers are concerned, phloem fibers are the only dead cells which are present in the phloem. Means they don't have the photoblast. They have the empty cavity in the center of the cell instead of having a protoplast. If there is no protoplast present there in the cell, we consider that cell as a dead cell. Okay, all these fibers present there in the phloem, they do not possess the protoplast. That is why we consider them as dead cells. As far as the very first type of cells which are there in the phloem are concerned, we call them as sieve elements. Okay, there are two types of sieve elements present. The very first kind of sieve elements which are there in the plants are usually called as sieve cells. The second type of sieve elements present in the phloem are referred as sieve tubes. Usually sieve cells, they are almost like this. They are elongated cells. Even somewhat cylindrical. These cells are usually the living cells. They have the protoplast. Okay. But here the end walls or cross walls are perforated. You can see the perforations there. Perforations means the small pore-like structures, the small perforations. You can see the cross walls or end walls. Okay. Through these perforations present in the cross walls, transportation of the food materials usually takes place. Food materials are getting transported from one cell to another cell almost through these pores. And these sieve cells are present there in the heterophytes as well as in gymnosperms. In these two groups of vascular plants, you can see the sieve cells. Sieve tubes are concerned. Sieve tubes are the advanced sieve elements. They are usually present in flowering plants such as angiosperms. When you look into the phloem tissue of an angiosperm in the longitudinal section, phloem more or less looks like this. You can observe the sieve tube cells like this. look at this. First of all, I am drawing the sieve tubes. These are supposed to be the sieve tubes. How they looks? They looks to be elongated and cylindrical shapes. They are very much elongated and it looks like the cylinder. These cells are generally having the crosswalks. This is supposed to be the crosswalk. And this crosswalk is usually referred as the sieve plate. I will tell you why it is referred as sieve plate. It looks like a plate having many sieves, means pores. When you look into the sieve plate, the sieve plate is having what you call as numerous pores on it, numerous perforations on it. Just like this. You can see the perforations. The plate-like structure having perforations can be called as sieve plate. Any structure having pores is usually called as a sieve structure. No doubt it is a plate-like structure having pores. That is why we refer it as a sieve plate. Plate-like structure containing pores or sieve. And another important thing that one should remember here is the sieve tube cells are usually the living cells. They are living in nature. And these living cells contain protoplast represented by the cytoplasm. Only cytoplasm is present. And that too, cytoplasm is going to be present in the form of what you call as strands. Means the fine fiber-like structures, just like this. These are supposed to be the plate-like structures. Okay, these plate-like structures are having what you call as the perforations. That is why we refer them as sieve plate. Suppose if you observe one sieve plate, it looks like this. Okay. You can observe the perforations. These are supposed to be the pores. And here every pore is covered with deposition made around the pore, and this deposition is going to form a pad-like structure. And the material that is going to be deposited surrounding the pore is supposed to be the callose. It is a kind of polysaccharide. And this callose is going to deposit surrounding the pore. Okay. We call them as callose pads. They are going to, or plugs even you can call. They are present surrounding what we call as the pores. This is the cytoplasm that is present, and this cytoplasm is present in the form of strands. We refer them as cytoplasmic strands. And these sieve tubes are enucleated cells. They are enucleated cells. Enucleated means without nucleus. These are the cells, they don't have the nucleus at all. In mature sieve tube cell of an angiosperm, You don't find the nucleus at all. They are without nucleus. That is why we call them as enucleated cells. These cytoplasmic strands, they are usually continuous from one suitube cell to another suitube cell, and these strands are going to pass through the pores. Because of their continuity from one cell to another cell, we also call these cytoplasmic strands as transcellular strands. We also call them as transcellular strands. And suitube cells are mainly meant for transportation of food. Okay, here no block is present. Remember, the end plates that are present in the form of sync plates, they are having numerous perforations. Through these perforations, transportation of the food molecules can take place. That is why we refer these cells as the food conducting cells of the phloem. Transportation of the food mainly takes place through these phloem cells, particularly the suitubes. By the side of every suitube cell, you can observe elongated narrow living cell. Can you write out here? This is supposed to be an elongated cell, and it is narrow cells. You can ask the question that what is this elongated and narrow cell? This elongated narrow cell is usually called as the Campanian cell. It is a Campanian cell for the suitube. It means that wherever there are suitubes present, by the side of the suitube always there is a cell associated with it. There is nothing but Campanian cell. And these two cells, suitube cell and Campanian cells, are originated from the same mother cell. Suppose assume that this is a prokaryote cell. Okay, I mean a meristematic cell. When it divides into two cells. Okay, it divides into two cells. Out of the two cells here, one is developing into what you call as a suitube cell. Another one is developing into what you call as Campanian cell. Okay, here this cell, if it becomes suitube, and this cell becomes Campanian cell. These two cells of the phloem tissue are usually derived from the same mother cell. They are ontogenically connected. Means they are connected from the time of their birth. Usually, Campanian cells, they have the cytoplasm. Even they too contain the nucleus. Even they have the protoplasmic connections with the suitube cells. Always between the Campanian cells and the suitube cells, living protoplasmic connections are present. We refer these living protoplasmic connections as plasmodesmata. Through these plasmodesmata, exchange of the materials takes place between suitube cells and the Campanian cells. They act as points of exchange. Okay, these points of exchange are usually the plasmodesmata. Even you will also find the thin walled parenchyma cells. Look here. These are supposed to be the thin walled parenchyma cells. Okay, these thin walled parenchyma cells which are present in association with the phloem cells. Either they may be linked to the suitube or they may be present by the side of the Campanian cell. We refer these cells as phloem parenchyma. Even you can also see the phloem fibers. These are the phloem cells. They are basically the fibers. They are the dead cells. They are present in association with the sieve tubes or the companion cells. Okay, we refer them as phloem fibers. Okay, this is how the phloem looks when you observe it through longitudinal section. Suppose if you cut the phloem in the transverse plane and observe the phloem in the transverse section, how exactly it looks. Phloem more or less looks like this. You can observe somewhat polygonal shaped cells. First of all, basically, you will see the cells like this. The phloem tissue is characterized by the appearance of one large cell, one small cell. Just like this. You may ask the question that what is this larger cell? Usually the larger cell is supposed to be the sieve tube cell. Okay. By the side of the larger cell, you will find the smaller cells. Okay. These smaller cells are usually the companion cells. These are supposed to be the companion cells. Okay. In addition to that, you can also see the sieve plate. Suppose if the section passes through the sieve plate, then you can observe the pore-like structures which are there in the sieve tube cell. Okay, it is supposed to be the sieve plate. Remember, and this sieve plate is present there in the sieve tube. Okay. You can see the parenchyma cells, phloem parenchyma. Okay. They may be polygonal or oval, sometimes rectangular.